board.
Thank you, Miron. Thank you, and welcome to everybody.
Yeah.
Thank you very much, Miron. Can you see my slides?
Yeah, everything can be seen. You can just change slides to see if that works. Just to make sure we are. Yeah, it works. Okay.
And you see it full screen?
Yeah. Everything is in full screen. We see everything, so you can continue. Thank you.
Perfect. So thank you very much, Miron. So I'm happy that the webinars are back, and today I'm gonna present let's say the very basics of gene regulation. So this is essentially gene regulation for beginners in terms of theory, of methodologies, and I will give you some examples from our work on how we're using gene regulation in order to answer specific questions related to cardiovascular diseases. So this is the presentation that I also gave in the training school, and the title is Transcriptional Regulation of Cardiovascular Gene Expression. The topics of my presentation will start with basic principles of gene regulation, promoters, enhancers, and transcription factors. Then we'll see some examples of transcription factors which are involved in cardiovascular diseases and their domain structures. Then we'll discuss some methodologies used to study gene regulation. And then we'll discuss about two examples, which will actually give us a good idea of why gene regulation is important to understand diseases and cardiovascular disease specifically. So one will be understanding the molecular base of cardiovascular diseases. This is a case study in which we analyzed mutations in the regulatory elements of ABCG1 gene, which are associated with ischemic heart disease. And the second example we'll see how we can use gene regulation techniques and transcriptomics in order to answer questions related to the differential gene expression in normal versus disease states. And we'll see the example of HNF4 and its role in non-alcoholic fatty liver disease. And we'll finish with some conclusions. So let's start from the beginning, some basic principles of gene regulation. So this slide actually shows in a very simplified form how a promoter looks like and how a promoter is, by what a promoter is occupied during transcription. So in this slide, I don't know if you can see my pointer. Can you see my pointer here?
Yes.
Okay, thank you. So in this slide, we see a typical promoter. A promoter is a region of approximately 100 to 200 base pairs next to the gene. So this arrow indicates the site where the transcription starts from a gene. This is called the transcription initiation. And just upstream of this transcription initiation site, there are regulatory elements such as these ones which bind sequence-specific transcription factors. We see some examples here, such as nuclear receptors, SREBPs, and SP1. Also, we see in this figure, we see the main enzyme which is responsible for transcription is called RNA polymerase II. RNA polymerase II is recruited on the promoter. It is placed specifically to the site of transcription initiation, and this binding is facilitated by auxiliary factors, which are called general transcription factors, or TF2s, TF2Ds, and general transcription factors, which interact with the polymerase and also with transcription factors. And this complex actually is very essential to correctly place the RNA polymerase II and start transcription from the correct site. Also, what we see in this figure is that in addition to the regulatory elements and the transcription factors, we see some other proteins which play very important roles. We see some coactivator complexes. Coactivators are enzymes, essentially, which bind to the transcription factors and help the chromatin to open in order for transcription to begin. This is accomplished by modifications of histones, as we'll discuss shortly. For instance, we see here a coactivator complex, is a histone acetyltransferase complex. And also, there are other proteins which are important for remodeling of the nucleosomes, because when transcription begins, the nucleosomes have to move relative to each other, and this is accomplished by specific enzymes which use ATP as an energy source. These are called remodeling complexes. In this next slide, we see essentially the same things. We see the gene, the RNA polymerase II, which has started transcribing. As you can see here, mRNA is produced. We see the data box, which is the region where RNA polymerase II and its, let's say, associated proteins bind. We see the mediators. And another thing that we see here is we see an enhancer. An enhancer is a region, a regulatory region, which consists of multiple regulatory sites, DNA binding sites, which is located further upstream or downstream from the gene. And usually what is happening is that the enhancer confirms tissue specificity or other specificity relative to the promoter. The promoter is used just, let's say, for basic transcription, but the enhancer can actually facilitate the tissue or conditional expression of the gene. And in order for the enhancer and the promoter to communicate with each other, we have a lot of protein-protein interactions between transcription factors and mediators, which bind to the promoters and to the enhancers. So in transcriptional regulation, we have a lot of interactions between proteins and DNA, and we have a lot of interactions between proteins and proteins. And there are many techniques which are very useful in order to characterize these interactions. In my lecture, I will focus mainly on protein-DNA interactions. There are many techniques for protein-protein interactions. I don't have time to cover these methodologies. I just briefly mention one, which is the histone-hybrid system to identify protein-protein interactions. Now, of course, the regulation of a gene depends heavily on the epigenetic modifications of its promoters and enhancers. As you can see here on the left, the histones are proteins which play a very important role in the organization of chromatin. There are different histones. They are called H2A, H2B, H3, and H4. And these histones make octamers, and these octamers actually are wrapped by DNA regions and make these nucleosomes. So each nucleosome consists of a histone octamer in the center and let's say a DNA region approximately 150 base pairs, which wraps around this this histone core. And the histones now that make this octamer have specific regions which are called histone tails, which are targets of the coactivator complexes which are present here, and they have enzymatic activities. So these histone tails can be modified either by acetylation or methylation or by phosphorylation. And these histone modifications, which are called histone marks, determine whether a specific regulatory region will be transcriptionally active or not. For instance, the acetylation of histones, what it does is it relaxes chromatin so that the transcription factors have access to the regulatory regions which are placed in these DNA regions. So if this is a promoter here, you see that this is the DNA of the promoter. These are the histones that wrapped around the promoter. So this region has to be open so that the transcription factors have access to the DNA binding sites. If this chromatin is compacted, such as here in this case, and here we have heterochromatin, then we have other modifications such as methylations, which are predominant in heterochromatin. And these histone marks are, let's say, they are markers of transcriptional inactivity. So depending on the mark, we can actually let's say, can predict whether a gene will be activated or will not be activated. So there are specific marks for active chromatin, and there are specific marks for inactive chromatin. And now we have the technology to identify these marks and determine whether the gene is active, or in general, the transcriptome, the chromatin of a given cell is active or not at any specific time. In order to see what marks are present in histones, we use a technique called chromatin immunoprecipitation that I will discuss in one of the next slides. Now, transcription factors are very important for gene regulation. These are the proteins that do the transcription. They bind to the DNA and they interact with RNA polymerase II and with the coactivators. And all these interactions actually are very important for transcription to start. A transcription factor consists of different modules. So it is a modular protein, and we have most of the transcription factors contain essentially similar modules, such as the DNA binding domain, which is important for binding to DNA, the transactivation domain, which is important to do the transactivation, meaning interaction with RNA polymerase II and transcription initiation. Nuclear localization is a region which is important for the transport of the factor after its synthesis to the nucleus. A ligand binding domain in certain transcription factors is important for binding of ligands, such as in nuclear receptors, and dimerization domain for cases where transcription factors act in dimers. So these modules are separated by hinges. So of course, each module has its own structure, well-defined structure. And these domains, these modules are separated by non-structural regions, which are called hinges. So each module can act in an autonomous fashion. So it's like the parts of a machine. Each part has its own function. You can take one part from one machine and you can put it in another machine and it can still work. And the same is here. You can have different modules which can be exchanged with modules from other transcription factors, and we can make hybrid transcription factors with different properties. And that was very important historically for the characterization of the domains of the transcription factors. The ability to move these domains from one protein to another was very critical in order to characterize these domains. So hybrid transcription factors are possible, let's say for structural functional studies. And this is the example that I mentioned, which is based on the ability of different modules to interact with each other. So this is let's say the famous yeast-2 hybrid system, which has been used to identify protein-protein interactions. And this yeast-2 hybrid system is based, as the name implies, in two hybrids. So we have a transcription factor from yeast, which is called GAL4. It has the DNA binding domain and the transactivation domain. So these are the domains. So this is the hinge that links these two domains. These are the two modules that I mentioned. So we can take one of these modules and link it to a different domain, which is called a bait, and take this domain and link it to another protein, which is called prey, and make these two hybrid proteins, express them together in a yeast cell, and see whether they interact or not. If they interact, if the bait interacts with the prey, this interaction will bring together the binding domain and the activation domain of GAL4, like this one. So we'll have this transcription factor restored, not via this hinge, but via these protein-protein interactions. So the bait and the prey play the role of the hinge that brings together the bait, the binding domain and the activation domain. So this example actually see the power of these modules, of these transcription factors to act as modular proteins, and how we can utilize this property in order to, let's say, study gene regulation and identify new things, such as protein-protein interactions, to see whether these two proteins interact or not inside the cell. Of course, there are techniques that we can use, in vitro techniques, with purified proteins to see whether the proteins interact. But this technique, the yeast-2 hybrid, was very popular because it is an in vivo technique. The interaction takes place in vivo, in a cell. Now, let's look at some examples of transcription factors and their domain structures. All the examples that I'm going to show you are from the cardiovascular field, but not only. Of course, there are no cardiovascular specific transcription factors. There are transcription factors which are involved in cardiovascular diseases, but not only. So one is the AP1 family of transcription factors. It's a large family. Two of the most, let's say, famous members are C-Jun and C-Phos. So this is the structural organization of a typical AP1 protein. This is C-Phos and this is C-Jun. So we have the DNA binding domain, which consists of basic amino acids. It's a DNA binding helix, which is shown here in purple here. So this is the DNA binding domain. And we have the leucine zipper domain, which is here, which consists of leucine residues which interact with each other and make this dimer. So one member of this family can interact with another member of this family via the leucine zipper domains, such as this one here, this dimer, consisting of C-Phos and C-Jun, and can bind to DNA's dimers. And this dimerization is very important for this family. So again, schematically, we see here C-Phos and C-Jun, the leucine zippers, the DNA binding domains, they make dimers, and they bind to DNA as dimer, and they interact with specific DNA binding regions. Now, these are sequence-specific transcription factors. They don't bind to DNA anywhere. They bind to specific DNA sequence elements. And what elements are these? For the AP1 family, the consensus recognition site for these proteins are TGA, CG, TCA. And so this is the specific sequence of nucleotides that must be present in a promoter in order for these family members to bind there. And how do we know that this sequence is an AP1 site? Because we have done experiments using the CHIP chromatin immunoprecipitation again, using a large number of promoters that are responsive to AP1. We have put them together, we have aligned them, and we have shown what nucleotides are the same in each position, and we have come up with this consensus. They are called consensus sequences. So this is an AP1 motif. It's called TRE, from TPA responsive element, because TPA activates these AP1 factors, and also cyclic AMP activates other factors of this family. These sites are called CRE, cyclic AMP responsive element. So a TRE has a sequence TGA, C or G TCA, and the AP1 motif, the CRE motif, has the sequence TGA, C, G TCA. Almost identical, but not exactly identical, very similar. Another family is the NF-κB. Most of you know NF-κB. It's a very well-studied transcription factor. Again, here we have a huge family with different members. We have dimers like the AP1. So we have proteins of the P50, P52 class. These are proteins that derived from the precursors called P105, the P100. And this is one subclass, and the other subclass are the REL A, B, and P65. And we have dimers between this subclass and this subclass. Again, the domain is similar, DNA binding domain and the transactivation or transrepression domain. Depending on the protein, a transcription factor can activate or repress transcription. The transrepression can be accomplished by interaction with transcriptional repressors, not co-activators, co-repressors, which are enzymes that do not do acetylation. Let's say they do histone deacetylation. As shown in this on the left, stimuli such as pro-inflammatory cytokines activate NF-κB, which is in complex with an inhibitor. The inhibitor, after the stimulation, is degraded, and the NF-κB goes to the nucleus. They make dimers, and they can either activate or repress transcription depending on the dimers. And again, you see here the structure of a dimer of P50 and P65, the dimerization domains, the DNA binding domains, and this is the consensus sequence for this NF-κB family. A third very important family is the family of nuclear receptors. It's a very big family compared to the other two. Here we have approximately 50 members. The structure contains the DNA binding domain, which is of the zinc finger type, shown here. It has a zinc that coordinates some amino acid residues and make this specific structural arrangement of alpha helices, which bind to the DNA. So this is the DNA binding domain. And we have the ligand binding domain that bind ligands. Ligand binding is very important for the activation of these proteins. And what is happening is that in the absence of the ligand, these nuclear receptors, which bind again as dimers, as the previous examples, they bind as heterodimers or homodimers, they actually do repression because they bind to repressors like the HDAC, histone deacetylases, which are called SMART or NCOR, nuclear receptor co-repressors. And the chromatin is inactive. When ligand binds, when ligand comes, let's say a hormone, and binds to these receptors, the ligand binding domain changes in conformation, and we have a new domain, a new ligand binding domain, and this change in the conformation displaces the co-repressors and recruits coactivators. Now, here, the co-repressors leave and coactivators come. The coactivators are histone acetylases. They acetylate histone, and the chromatin opens, so we have transcription initiation. And there are very famous proteins here in this family, including the classical hormone receptors, such as glucocorticoids, estrogens, androgen receptors, etc. We have some factors which are very important for cardiovascular diseases because they are involved in cholesterol and fatty acid biosynthesis, such as the epipars, the LXRs, FXRs for bile acids, RXRs, and we have a class which are orphan receptors, meaning that for this specific subfamily, we don't know the ligand, or maybe there is no ligand, and these are constitutively active. In this third subfamily, we have HNF4, for which I'm going to talk to you at the end of my presentation. And again, these transcription factors are regulated. They do not act constitutively. They are activated by signal transduction pathways. And here in this slide, we see some signal transduction pathways which activate these transcription factors. For instance, cytokines such as TNF-alpha or interleukin-1, they can activate NF-κB, can activate genes which are targets of NF-κB. Cytokines can also activate different factors for which we did not discuss, but again, they have similar structure, the STATs, by activating the Janus kinases and the STATs, transcription factors. We have seen the AP1, the Jun and C4s, bind to DNA and activated also by cytokines. And we have also here is the AP1, here is the NFAT is another signaling pathway, and we have also the AP1. In all of these cascades, we have a series of phosphorylations of kinases. One kinase activates the other, and then the signal actually goes to the transcription factor, which could be, let's say, phosphorylation or nuclear translocation, some modification at least, and then the factor is activated. And we saw also the case of steroid hormones, steroid receptors, which are activated by steroid hormones. Now, of course, promoter gene regulation is not simple, it's very complex. And just to give you a glimpse of the complexity, this is a figure showing the regulation of one gene, which is very important for HDL metabolism, and this is the ABCC1 gene. And this is one figure that we made with Anka Gafenko some years ago, when we wrote a review for this book, High Density Lipoproteins. And this actually figure summarizes all the transcription factors and all the signals that regulate the expression of ABCA1. You see, there are signals that let's say, result in the activation of certain transcription factors. So this is very complicated. The question is, how do we study this? How do we identify the transcription factors, and how do we identify the proteins that regulate these genes? So that brings me to the next part of my presentation, which is the, let's say, the practical part. I'm going to give you some techniques, discuss about some techniques, very few, not too many, the very basics that we use in order to study gene regulation. So the first technique that everybody in a gene regulation lab uses is the luciferase assay. In this assay, you measure the activity of the luciferase enzyme, which comes from the fireflies, produces light. We know the fireflies in the dark, they produce light. The reason they produce light, these fireflies, is because they express luciferase, and the luciferase enzyme catalyzes this reaction. It actually oxidizes luciferin, which is the substrate, with the help of ATP to oxyluciferin plus AMP plus pyrophosphate plus CO2 plus light. So this is the reaction that produces light that we can measure using an appropriate instrument, which is called the luminometer, which actually measures bioluminescence. So what we do is when we want to study, let's say, the promoter or the enhancer of our gene, we take this promoter, we clone it from human genomic DNA or any genomic DNA, and we clone it upstream of this luciferase gene here in this let's say, multiple cloning site. We use these restriction sites and we insert our promoter here. Once we make this plasmid, which is called the luciferase vector or the reporter vector, containing our promoter upstream of the luciferase gene, we take this plasmid and we put it into bacteria, into cells, into eukaryotic cells with a method that is called transfection. So what we do is, here is the reporter. We make a transfection mix consisting of a DNA and other, let's say, transfection reagents, such as lipids, liposomes, or calcium phosphate, depending on the technique. There are many techniques for transfection. And then this transfection mix is placed on top of the cells. So we add it to the medium. This will sit on top of the cells, and the cell will take it up, will phagocytose these transfection mixes, and all the DNA that we put in the mix will end up in the nucleus and will be expressed. And now here, I'm showing you the same technique, the transfection, in which you can co-transfect two different plasmids. One is your reporter, and the other is another plasmid which expresses a transcription factor, overexpresses a transcription factor, this is called an expression vector. So you can do this co-transfection technique. You can transfect these two plasmids at the same time and see whether the transcription factor that is expressed from this vector will go and activate your promoter in the other vector. And this will happen inside the nucleus. So if this protein expressed here is a transcriptional activator of your promoter, we'll see more luciferase when we analyze the extracts from the cells. Okay, so we do the transfection, we put the DNA into the cells, we leave the cells for about, let's say, 16 to 24 hours, and after that, we lyse the cells, we take the extract, and we do the luciferase assay. Now, in order to study protein DNA interactions and see whether a specific regulatory region binds a transcription factor, we can use two techniques. One is in vitro and the other is in vivo. The in vitro technique is called DNA affinity precipitation, in which we actually make a PCR product containing the regulatory region of our gene. So if this is our promoter, and here if we suspect that here there is a binding site that resembles, let's say, the consensus site of a transcription factor, we amplify this region using primers by PCR. We biotinylate this primer, so the PCR product is biotinylated, and we take this PCR product, we incubate it with nuclear extracts from the nuclei of cells that we know that express a transcription factor, and then we incubate these extracts with the biotinylated DNA, and then we purify the DNA using magnetic streptavidin beads, because streptavidin binds to biotin, which is on the DNA. And so we precipitate the DNA actually, and if our nuclear factor was bound to the DNA, will co-precipitate. And we take these precipitates and we run SDS-PH and Western blot using an antibody for a transcription factor and see whether our transcription factor is present in the precipitate. I will show you some examples in the first case study for these techniques. And finally, this is the chromatin immunoprecipitation, which has been widely used. Once it was discovered many decades ago, it was widely used in gene regulation to identify protein-DNA interactions in vivo, and also to identify modifications in chromatin, such as the histone modifications. Chromatin immunoprecipitation is very important to characterize the histone modifications and see whether a gene is, let's say, has marks for transcriptional repression or transcriptional activation. So the basic principle of this technique is that we take a cell line, which is cultured in a Petri dish, and we add formaldehyde, and we fix all the interactions that are present inside the cell, protein DNA and protein-protein interactions. Everything is fixed with formaldehyde, and then we lyse the cells, we isolate the chromatin, the DNA, and we sonicate the chromatin so that we cut the chromatin into small pieces, because it's very difficult to work with unsonicated chromatin. It's very viscous, so we cut it to pieces, and then we add an antibody against a transcription factor that we are studying. So this antibody will recognize the proteins that are bound to the chromatin when we added formaldehyde in this time point here. So if there is a protein that was bound to our promoter, and if that protein was our transcription factor, then using an antibody for this transcription factor, this antibody will bind to this protein bound to our promoter. And we do regular immunoprecipitation. The factor will be immunoprecipitated, but because it is covalently linked to the DNA will be immunoprecipitated as well. So in the immunoprecipitates, which consist of chromatin and DNA, then we get rid of the chromatin because of the proteins. We want we're interested in the DNA that came with this immunoprecipitation. So we take this chromatin with a reverse cross-link, we purify the DNA, and we do PCR with primers specific for our promoter. So if we see a PCR fragment, it means that our promoter was present in the immunoprecipitate, meaning that this transcription factor was bound to our promoter when we added formaldehyde here. Very important tool, very important also for you can actually scale up this technique by, let's say, sequencing all the chromatin fragments that are immunoprecipitated. This is a version called chromatin ChIP-seq, chromatin immunoprecipitation sequencing. Instead of, let's say, focusing on one gene, you can sequence everything and see what regulatory regions one transcription factor binds. Now, I will go to the first case study. And in this case study, we'll see why gene regulation is important to understand the molecular basis of diseases. So here we'll see an example where we characterize the mutation in the regulatory element of a gene, which and these mutations are associated with ischemic heart disease. So there are not many papers like this in the literature. So I had to go back and select one of our previous papers, which was published quite long time ago. It was almost 12 years ago. That was a collaboration with Anne Timber-Hansen from Copenhagen. It was published in ATVB, and the title was Genetic Variation in ABCG1 and Risk of Myocardial Infarction and Ischemic Heart Disease. So in this paper, we will see how we use these techniques in order to characterize mutations, functional mutations in the regulatory regions of genes, which are associated with ischemic heart disease. First of all, for those of you who are not in the HDL field, ABCG1 is a lipid transporter which are expressed in many tissues, but is also expressed in macrophages, which play important role in atherosclerotic plaques. So this is the ABCG1 gene. It's a transporter. It is activated by cholesterol and its hydroxylated derivatives, such as which are called oxysterols. So ABCG1 plays an important role in the so-called reverse cholesterol transport pathway of HDL, because it takes cholesterol from the macrophages and transfer this cholesterol to HDL particles, primarily to mature HDL particles, for transport back to the liver and excretion. So that was the study plan. First of all, our collaborators in Copenhagen, which did the genetic analysis, we did the functional work on the promoter. So in Copenhagen, they did next generation sequencing of the core promoter, coding regions, and splice sites of ABCG1 in individuals in the general population of Copenhagen. This is called the Copenhagen City Heart Study, with the lowest 2% and the highest 2% HDL cholesterol levels. That was the first step. The second step was the genotyping of 10,000 individuals from the general population, not the Copenhagen City Heart Study, the general population of Copenhagen, for the variants identified, to see whether these variants can actually predict whether one person will have ischemic heart disease based on these polymorphisms, and determine the association with plasma levels of HDL cholesterol and other lipids and lipoproteins, and the ability of these variants to predict risk of myocardial infarction and ischemic heart disease with 34 years of follow-up. So they want to see whether these variants can use as genetic risk factors, genetic predictors. And the step 3 was another validation using another cohort, now consisting of patients who have myocardial infarction, ischemic heart disease in Copenhagen City Heart Study. So 3 steps to find genetic variants that can predict ischemic heart disease, but variants in the regulatory regions, not in the coding regions. So the focus of this paper was on the regulatory regions of the genes. We're not talking about mutations in the protein. We're talking about mutations that affect the regulation of this gene. So this is a figure showing all the different polymorphisms, so the different variants. So this is the genotypes. This is the effect of these variants on HDL cholesterol levels. All these variants, the minus means that they are upstream of the gene, they are not in the coding region. And you can very clearly see that none of these variants in the regulatory regions affect HDL cholesterol. There's no statistical difference between, let's say, patients that had one variant, one allele versus the other in terms of HDL cholesterol. The p-values were very large. But one of these variants, this one, the minus 376C to T, had, let's say, a hazard ratio for myocardial infarction, this one, which predicts ischemic heart disease. So this genetic variant was strongly associated with myocardial infarction and ischemic heart disease in the Copenhagen City Heart Study. Only this variant from many different variants that were analyzed. So it was so these results prompted us to investigate whether this variant here was a mutation, not just a variant, let's say a silent polymorphism, but indeed a real mutation that can cause the myocardial infarction and ischemic heart disease. So we collaborated with Antibody Hansen. They sent us the information, and we cloned the promoter of the ABCG1 gene. Actually, that was upstream of exon 5. The regulation of ABCG1 is quite complex. It can start from many different regions, but this was the promoter upstream of exon 5. So we cloned this promoter, and this is the polymorphism, the variant minus 376C to T. So in this position, we can have either C or T. Now, using bioinformatic tools, you can see whether a specific DNA region contains a binding site for a transcription factor. So there are specific bioinformatic tools that use all these consensus elements that are published. They put them together and find homologies with the consensus sites. And for this variant, it gave us homology with a binding site for a transcription factor called SP1, specificity protein 1, which has multiple Cs and Gs. This is the consensus binding site for SP1. So if you compare this region, this sequence with the consensus, there is some homology. And the T now, the presence of the T somehow changes this consensus, changes the binding site, presumably. We didn't know that. We wanted to prove that. So then after we cloned the promoter, we mutagenized this C to T. So we made two constructs, which are shown here, the C allele and the T allele. And we did transfections, as I explained, in cells, in cells. Now, the cells that we chose were not chosen randomly. There were specific cells from Drosophila which do not express SP1 at all, so that the background is 0. That was very important because SP1 is very ubiquitous and it's very abundant. It's expressed everywhere. So we wanted a cell to have zero background of SP1 so that we see the effect of overexpression of SP1 in these cells. And what we did was we transfected these cells with the promoters, this one or this one. So this is the C allele, this is the T allele, this one and this one. And this is the activity. It was very low anyway in Drosophila cells. But when we co-transfected SP1, so SP1 was expressed from this plasmid here that I show you, this one, SP1 here, the reporter vector here, co-transfection, luciferase assay. What we saw that when we overexpressed SP1, this promoter was activated very strongly, whereas the other promoter containing the T allele was not activated so strongly. It was still activated, but not as strongly as this one. We also used other members of this family, SP2, SP3, and we did not find anything. So SP1 was, based on this data, SP1 is a transcriptional activator of the ABCG1 promoter, and a mutation in this region affects the transactivation potential of SP1. Does not diminish it, because there may be other SP1 sites that are present, which can also work, but this reduction strongly indicates that this site is indeed an SP1 site. Now, of course, we want to prove that SP1 indeed binds to this region. We use the DNA affinity precipitation that I already described. We purified this, we amplified this region with primers, biotinylated primers, and we saw that we incubated with nuclear extracts from cells, and we found that SP1 binds very nicely to the -376 allele, but does not bind to the -376T allele. You see that the band here in the Western blot is absent. Here, the Western blot was probed with an antibody for SP1. And this is the positive control using another region from ABCA1, another transporter that binds SP1, and a negative control, the data box region of ABC1 that does not bind SP1. So this in vitro data confirmed that SP1 binds and the mutation affects the binding of SP1. And a final experiment that we did more in vivo was a chromatin immunoprecipitation, where we did the experiment, the protocol that I described, and we immunoprecipitated the chromatin with an SP1, and we found that the ABCG1 promoter was there, as you can see here. This is SP1 binding to ABCG1 promoter. But when we added a drug in the cells before formaldehyde, we added mithramycin A, which is a known anti-cancer drug, which intercalates, causes DNA damage, but it intercalates in regions where there are many Cs and Gs, like the SP1 site. Mithramycin intercalates into these regions and does not allow the transcription factor to bind. In other words, mithramycin is an SP1 inhibitor because it does not allow SP1 to bind. And that was known, it was previously published. So we use mithramycin, and this drug also reduced the binding of SP1 in vivo. So that was a very another good indication. So in conclusion, this was the first report of a functional variant in ABCG1 that associated with an increased risk for myocardial infarction and ischemic heart disease in the general population without a correlation with plasma HDL or other lipoprotein levels. So it was very interesting and very crucial. and very strange, of course. And the study required promoter cloning, generation of luciferase vectors, in silico analysis of transcription factor binding sites, site-directed mutagenesis, transient transfections, luciferase assays, DNA-AP assays, and chromatin immunoprecipitate assays. And finally, in the last, let's say, 10 minutes, I will go quickly to a second case study. In this case study, again from our work, actually will demonstrate the importance of using gene regulation techniques such as transcriptomics or real-time PCR, because the real-time PCR and transcriptomics are part of the gene regulation field, because they study the expression levels of specific genes and how these levels change in response to stimuli. And all these stimuli, of course, change transcription factors, which activate these genes. And here we see an example of what is happening when a transcription factor is affected, and that can cause a disease. The transcription factor is HNF4, and the disease is non-alcoholic fatty liver disease, which is actually a quite frequent disease. Fatty liver disease actually can be found in approximately 20 to 30% of the population. It is characterized initially by lipogenesis, increased lipogenesis due to dyslipidemia, insulin resistance, obesity, metabolic syndrome, etc. And then if this situation here is untreated, and it is very difficult to be treated, because this disease often goes undiagnosed, because it may not be accompanied by liver enzyme elevation. So it's difficult to identify people in the population that have fatty liver, especially when this is in the first stages, in order to prevent the, let's say, the conversion to more advanced stages, which are more severe, such as the non-alcoholic steatohepatitis. So this is a non-alcoholic fatty liver disease. 10 to 20% of these NAFLD patients become NAS patients, non-alcoholic steatohepatitis, that have, let's say, lipid peroxidation, apoptosis, oxidative stress, mitochondrial dysfunction, and 2% of these patients will end up with cirrhosis, advanced cell damage and scarring. So this is severe disease, can go unnoticed, and as the sooner you treat it, the better. The problem is that there are no drugs to treat this disease. And the fatty liver disease is the main characteristic is the accumulation of lipids, primarily triglycerides, in the liver. And this accumulation of triglycerides, of course, is not normal. It causes many problems, causes liver dysfunction. The liver does not function very well, and it can be caused by the dysfunction of transcription factors, which are called nuclear receptors. For instance, when we have superactivation of PPAR gamma, we can have increased fatty acid uptake and lipid droplet formation, because these are the target genes of PPAR gamma. All these are nuclear receptors in red. LXRs can increase de novo lipogenesis, and you have increased triglycerides when it is activated, let's say by high-fat diet. Or if you have the activation of PPAR alpha, you have decreased fatty acid oxidation. Again, triglycerides accumulate, they are not catabolized. And when you have defects in lipoprotein secretion, then again, you can have fatty liver, because the lipoproteins are not secreted, they stay in the liver. And this is the case with HNF4. So HNF4 is reduced in us. We know that from humans and mice. So in humans, the mRNA levels of HNF4 are reduced at the mRNA and also at the protein level. And in mice, if you treat mice with high-fat diet and you have fatty liver, HNF4 is reduced. So we asked the question, is there a causal relationship between low HNF4 levels, as you can see here, and NAS? And of course, to answer this question, you need to have an in vivo model. And what we did was we generated an in vivo conditional knockout model, mouse model of HNF4. We used albumin CRE mice that express CRE recombination in the liver only. We crossed these mice with floxed mice that contain HNF4 surrounded by LOXP sites. So this crossing generated mice which are conditional knockout for HNF4. That is, the gene is not expressed only in the liver that expresses the Crete recombinase. And we found that these mice, they have, of course, they do not express HNF4 at all in the liver. It expresses naturally in other tissues. And if you look at the liver is the weight of the liver is larger, as you can see here. So this is the liver weight versus body weight in female and male mice. This is the knockout, and this is the controls. And of course, you can see by the eye, by looking at it, that the liver is fatty because it has this pink color compared to the normal liver. And of course, if you do histology, you find all these clinical features of NASH. And just to make a long story short, I will not go through any details. All I want to show you is that using appropriate tools and techniques, such as the knockout generation of the conditional knockout model, the real-time PCR, and the transcriptomics, you can answer questions related to the effect of, let's say, altered expression of a gene in a disease, in a global scale, not in a gene-specific scale. So in the ABCG1 case, we studied only ABCG1. Here we study globally what is happening in the liver when a transcription factor either is not present or is downregulated. So using these techniques, we can ask more global questions. And of course, these experiments gives you a new information so that you can open new projects and generate new questions regarding the role of the genes that you are going to identify. So how can you identify new genes that are involved in an AFLD, having established an AFLD? So you see here we have a model of an AFLD. Now we can take these tissues and you can see how many genes change. And this you can do either at the single gene level using real-time PCR, as you can see here for many apolipoproteins. All apolipoproteins are affected by the HNF4 deletion, or by plasma enzymes. You see essentially all the plasma enzymes are also affected. And you can do at the global scale using transcriptomics, either microRNAs or RNA sequencing. We used microRNAs in that study to compare the transcriptome of the liver transcriptome in the two, in the knockout and the controls, and we found many genes, and these are the bioinformatic analysis of this data showing all these diseases and pathways which are affected in the liver, in the HNF4 liver knockout mice. And of course, we identify fatty acids, cholesterol metabolism, PPAR signaling, and many others. And using bioinformatic tools, you can actually see what is happening in terms of gene networks. You can identify networks of genes which are affected, and then you can study one network at a time. If you are interested in this, and you have, of course, the money and the power, you can actually study these pathways and validate these pathways, because all the transcriptomics and the bioinformatics just give you the information. Then it's up to you to validate these findings or leave it there for someone else. Let's say deposit the data somewhere in a database, and someone else from another lab may go mine them out and analyze them. And this is just a summary of the transcriptomics data showing what is happening in this mouse model of fatty liver disease, which genes are affected, which transcription factors are affected, and how the lipoprotein metabolism was affected. And this is the conclusion. So this is my final slide. So gene regulation in the cardiovascular system is a field of intensive research for many decades. Many transcription factors were identified to play important roles in atherosclerosis pathophysiology, as well as drug treatment. Some well-studied examples of transcription factors as the NF-κB, AP1, the hormonuclear receptors. These are the examples that have been studied very thoroughly. Gene regulation can be studied using a panel of in vitro, ex vivo, and in vivo methodologies, and new, more advanced technologies are being discovered every year. Understanding the mechanisms of gene regulation and transcription factor regulation is crucial for understanding the molecular basis of cardiovascular diseases, differential gene expression in health and disease, and of course, understanding drug actions, because most of the drugs that are in the market, they affect transcription factors such as hormone nuclear receptors, for instance, or NF-κB and other transcription factors. So it's important to have a knowledge of how these transcriptions work in order to understand drug actions and actually develop new drugs. So that's about it from me. I can stop here. Let me stop sharing.
Thank you so much, Dimitri, for this insightful lecture and really emphasizing all the aspects, also from the theoretical side, what is important, but also going more into details about the analysis and really providing us with all the steps that are not easy to take, but they are necessary if you want to analyze all this. So we can start with the discussion. I don't know if we have any questions from the audience. Paolo, do you have any questions at this point?
Yeah, I only have a comment because, you know, I like very much the translational approaches. So Dimitris, I like very much the fact that you started validating, let's say, SP1 activity from a clinical study. So you use the basic research as the magnifying lens for a phenomenon that is observed in a population, or vice versa, you can replicate in a model, mouse model or whatever, or cell model, a specific disease. So this is very important to me because we can utilize the basic research models as tools in any condition after a clinical evidence or molecular evidence of something, or before or in parallel to highlight some pathways. So I guess this is the message. So your seminar actually really highlighted this picture. So the great flexibility of these tools.
Yes, thank you. And actually, unfortunately, this validation of variants is not something that you see often in the literature. You see many genetic studies identifying variants, associations with diseases such as ZWA studies, etc. But you don't see functional characterization of these variants to identify the ones that are real mutations or just variants and the mutations somewhere else. I think it's very important to see what the mutations are. And if you want to understand what this patient has, you have to characterize the mutation and see whether this mutation causes the disease or not. Maybe it doesn't. Maybe another mutation somewhere else, genetically linked with that one. But I mean, if you identify the mutation, then you can try to see whether you can capitalize on that and make a drug. To let's say, to activate a transcription factor that is not binding very well, maybe to activate it more in order to make it bind very better and so on. So it's important information, but unfortunately it's missing from the literature.
It's a matter of improving the collaboration between, let's say, clinicians or molecular clinicians, if you want, and basic researchers with these tools.
Exactly.
So this is something to maybe to implement some projects, some grant application connecting the two ways.
Yes, exactly.
Yes, great, very interesting.
Yeah, also comment from my side, I think that what you just emphasized, Dimitris, is really important because we have many GWAS studies done and we have many SNPs and mutations that are out there and showing us that there is some relations, but not many of them are really analyzed in depth like you did with this one that you showed us. And I think it's really important to focus in the translational research on this part, really connecting both worlds.
And the reason that we were involved is that these collaborators of ours, they analyzed the regulatory regions, right? The coding regions, of course, is a different game. It's not the same. If you try to characterize mutations in the coding regions, then you have to clone the gene, and then you mutagenize, and then you have to make, let's say, put it in mice. We have done that for other mutations in collaboration with Anne, with adenoviruses, for instance. You infect the mice with viruses containing mutant genes, but this is another ballgame. In this case, we're lucky that they focused on the regulatory regions. That's why our expertise was important for this collaboration.
Yes, thank you. Do we have any other questions from the audience? I don't think, let me just check if I see any hands raising or something, but I don't think so. If that's the case, I can start with my lecture, and maybe later on we can continue with comments on the both sides. I will now
Should
share
we
my...
introduce you or not?
Sorry?
Should we introduce you or not? I mean, we
yeah, I mean, if you
anything.
want, but I think that at least for this audience, I'm pretty, they know who I am, so I don't think that I need any more introduction. I'm the guy who sends all the emails, so.
Exactly.
Well, not a little bit more, I would say.
Currently, it's just that. Currently, it's just that. Okay.
Miron, I meant the man of the spreadsheet. No, I'm just kidding.
That's like Dr. Jekyll and Mr. Hyde. Dr. Jekyll is one who sends emails, and Mr. Hyde is the spreadsheet guy.
No, substantial contribution to all our work. Let's say this, substantial contribution to the scientific work. Thank you so much.
Thank you so much. Okay, can't you see my slides? Let me just check if I can change them. Yes, it works.
Yes.
For me. Do you see the changes in the slides and everything? Yes. Okay, perfect. So as we discussed in the training school, the original title of the presentation was Post-transcriptional Regulation of Gene Expression, with the focus mostly on microRNAs and other types of non-coding RNAs. But since many of these RNAs are also regulating on transcriptional level, I also put a little brackets here on this part, which is post in the title. So I would like to start from the beginning and from basically what we call the central dogma is the story that we were taught many years ago, that basically we have genes in our DNA, and DNA is going to be translated to the RNA, mRNA, and then mRNA is going to be translated into proteins. And basically that was like a straight line from gene to proteins. But since then, we learned that there are many underlying mechanisms that control this process and really fine-tune how this story develops, the story from gene to protein. And this can also be seen in the numbers because we have around 25K of structural genes in the DNA. We have about 25K of transcripts, which is 10 times more. And when it comes to proteins, I always try to find what is the number of proteins, but the literature is really, really diverse in the providing me answers. So sometimes it's 500K, sometimes it's 1 million. So it is really evident that there are many mechanisms that complicate the story from gene to protein. When I said complicate, I mean they regulate on many different instances this roadmap from the gene to the protein. And of course, some of these mechanisms were mentioned by Dimitris previously, and also I will try to focus on the other mechanisms. One crucial part of my story is going to be dedicated to so-called non-coding RNAs, and this is something that in literature sometimes they refer to as so-called dark side of the genome, in reference to dark matter and dark energy in cosmology. And if you see actually this graph that was produced by CardioRNA, our previous cost action, you can see basically the relationship between genome transcriptome and coding transcriptome. So basically you can see that 80% of transcriptome is non-coding RNAs, and only maybe 2 to 5% is coding RNAs, which basically this is the part of the genome that produces the mRNAs. Of course, when we look at the transcriptome in more detail, we will see in this non-coding part, we will see some players, some types of RNAs that are very well described and recognized, and like transporting RNAs and ribosomal RNAs. But also we have a portion of RNAs that had been discovered in previous 2 decades, and they were really analyzed and deeply described in previous 2 decades, like small and long non-coding RNAs. And within small non-coding RNAs, again, we have microRNAs, we have pRNAs, snRNAs, SNRNAs, and within long non-coding RNAs, we have link RNAs and circRNAs and other types of non-coding RNAs. Just to emphasize the meaning of these non-coding genes and how they are actually related to the complexity of the organism. I always use this representation when on one side I put Homo sapiens and on the other side C. elegans, which was recently really put into focus because these are the ones that were used to discover microRNAs. And as you probably heard, this year a Nobel Prize was given for the discovery of microRNAs. And as you can compare both types of species, they have relatively the same number of coding genes, and the same number of non-coding genes for that matter. But when you compare the distribution within these non-coding genes, you will see that in organisms that are less complex, you have many of these non-coding RNAs or genes that are very small. And on the other side, when you have more complex organisms, you will see the increase in long non-coding genes. Also, we should not really represent the story of pseudogenes, which are also very important because you can see that as the organisms get more complex, the number of pseudogenes increases. So within this lecture, because it's very limited on time, I will focus my discussion on the three types of RNAs that are mostly and studied in particularly in cardiovascular disease field, like microRNAs, long non-coding RNAs, and circular RNAs. So microRNAs are, let's start with those that are best known and maybe most famous one. So microRNAs, the title itself, the name of the type of RNAs, I think describes well what do they look like. They are very short, about 20 to 25 nucleotides long. Just to put that in a perspective, you know that mRNAs are mostly above 200 nucleotides long. And there is a limited number of microRNAs in comparison to, again, mRNAs. As I mentioned earlier, there is around 20,000 coding genes, and here we have only 2,600, again, only quote-unquote microRNAs. These type of RNAs are very well conserved across species. So if you compare the sequences of microRNAs and their function, to that matter, in organisms like, I don't know, mice and rats, and then try to translate this to humans, you will see that the sequences are very conserved. One thing that complicates our interpretation of microRNA functions is that the fact that one microRNA can have multiple mRNA targets, and then again, several microRNAs can target the same mRNA. So this really complicates the interpretation of the function of certain microRNAs. So in the terms of their activity, they are not super specific. Their place of their activation is basically untranslated, 3' untranslated region of the mRNA, but it's not exclusively there. Most of them are depending on the activity on the 3' UTR, but to be honest, there are a certain amount of microRNAs that can also act on coding and region and 5' UTR. When it comes to the tissue distribution for microRNAs, again, there is low tissue specificity, which means that they are mostly present in different tissues. Of course, there are some microRNAs that are more present in one type of tissue. For example, in the context of cardiovascular disease, what you might find interesting is microRNAs that are called myomeres, which are super enriched in like cardiac tissue and generally speaking muscle tissues. But most of them are present everywhere. So when we say enriched, what I mean, there is more of them within this within specific type of tissue, but again, they are not exclusively present in only one type of tissue. Also, they are relatively different in quantity. So when you measure them in blood, some might be very well expressed and present in bodily fluids, but other might be very low expressed. They are relatively druggable. You can create either mere mimics, so to synthesize the microRNA and then administer it and use it as a drug, but also you can block certain microRNAs using antagomers, which is basically the anti-sequence of the specific microRNA. As I mentioned, they are present in body fluids, and they are stable there. But again, they are very, very often discussed as a tool that can be used as a diagnostic tool or prognostic tool because they have certain biomarker potential. But the fact that there is that they are not super specific regarding tissue distribution makes them relatively hard to really put in the biomarker context. So in that case, probably what you will see in different papers that they combine different microRNAs to get a model that is better diagnostic or prognostic tool. So let's deal a little bit with the biology behind microRNAs. So most microRNAs in our body are synthesized, are coming not for specific designating genes for microRNAs, but they basically come from introns of other coding genes. Of course, to a certain extent, there are some microRNAs that are coming from what we call microRNA gene or designated place in the genome for this microRNA. In any case, they can be transcribed with the activity of RNA polymerase 2 or 3, depending on the microRNAs, and then you get this precursor, which is called pre-mRNA, which is a very long hairpin look transcript. And then in the nucleus, this transcript undergoes activity of this complex, which is called DROSHA DGCR8. And then what this complex does, it basically cuts this pre-mRNA, and then you get something that is called pre-mRNA, which is a little bit shorter version of this transcript. Then it gets exported through specific type of transporters outside the nucleus into cytosol, and there it becomes a substrate for a specific nuclease that is called dicer. Now, dicer is really important because you can see here it removes this loop of the hairpin, and then what you have is basically two strands of RNA that are duplexed together. And also, please just note it, I will pay attention to this later, but there are these overhangs on these sides, and this is very important. And then microRNA gets loaded to what is called the RISC complex, and basically through the activity within the RISC complex, it exerts its activity. What RISC complex does, it enables microRNA to bind to specific target on the mRNA, and then basically it causes the gene regulation. So this part is actually devoted to the gene regulation. Now, how does this work? Basically, a RISC complex can induce two types of effects on target mRNA. It can cause translation repression, which means that ribosome will be bound to the mRNA, but due to the physical blockage of this RISC complex, the translation will not happen. So you basically don't destroy anything, you just stall the translation. And the other path that this story can take is mRNA cleavage, where basically when RISC complex binds to target mRNA, it causes mRNA to be completely destroyed. Now you may ask me, okay, Miron, how RISC complex knows which of these two scenarios are going to take place? Is it random? Can we know a little bit more about these two different outcomes? Well, it all goes to the, I would say, compatibility or the level of sequence compatibility between miRNA and its target mRNA. Because as I mentioned earlier, miRNAs are not super specific for their targets, because we see that there are 2,600 miRNAs that target around 20,000 genes. So by pure mathematical calculation, you can see that every miRNA can bind to multiple targets, which means that there is no 100% compatibility between microRNA and its target. However, two scenarios are possible. If there is like 90% of the compatibility, then this scenario will happen: cleavage of mRNA and its total destruction. On the other hand, if this compatibility is there, but it's not 95%, it's less, then the first scenario is going to happen. Now, I forgot to mention that within this duplex, you have two parts. One part is called 5P and the other one is called 3P, or 5P arm and 3P arm. And one of these, only one of those will end up being in the RISC complex. The other one is going to be released from the RISC complex and is going to be destroyed. So the question arises, is it always the 5P that is going to be inside the RISC complex? Is it always the 3P? Or again, is it something random? Is it something that we can understand how it's happening? And I will come back to this on the next slide. Just remember that there are these two parts. And of course, every time the strand that gets within the RISC complex, it's called guide strand. And it can be sometimes 5P, sometimes 3P. And the other one is called passenger strand. And this one is going to be degraded. So before I continue with the story of the 5P and 3P arm and how this is how the system chooses which one is going to stay in the RISC complex, I would like to give you a little heads up about the microRNA nomenclature, which for me is quite a complicated story. When you start working with microRNAs, first things you notice that names looks very, very random, and there are very similar microRNAs, but then again, you will see that one has this function and the other has that function. So I just want to emphasize a little bit about the nomenclature of microRNAs. So for this one, I use this example, which is microRNA 92. And as you can see, this microRNA can basically be produced from three different locuses in our genome. Also, please look at the part B of this figure, and you will see three different transcripts. And pay attention to this, what is called seed sequence. Seed sequence is the sequence that actually recognizes target sequence of mRNA. So when it comes to the function of mRNA, the seed sequence is the one that is actually the most important one. And since all these three transcripts have the same seed sequence, they are basically numerated as the same microRNA 92, because the seed sequence will actually tell you what is your microRNA. And in this case, all three types of microRNA have the same seed sequence. Then, in order to understand all the other letters and the nomenclature, we need to understand go a little bit back and understand how these microRNAs are produced. So you can see here that these two microRNAs that are called microRNA 92A are coming again from two different genes, but in the end, they are called the same, microRNA 92A3P. So why do they have the same name? Why do we call them with the same? Why do we designate the same type of name for them? You can see basically that if you look the sequence of both, they are not exactly only in seed sequence, but in the entire sequence, which means that they are the same. If you want to be super specific about their origin, then you can designate this one or two, which basically tells you that one is different from the other, but not on the final transcript level, not on the microRNA level, but on the pre-microRNA level, because they are coming from two different genes. At this stage, they are a little bit different. So this is why we gave them number 1 and 2. And this A thing really emphasized that the complete structure of the entire RNA is the same. And if you look the other or the third one, you will see that it's called microRNA 92B. And this B emphasized the fact that there are some changes in the structure that is not a seed, not the structure, but the sequence that is not a seed sequence. So you can see this asterisk here that really gives you the idea that there you see changes. So this is why we call this one 99B. Now, if we had another microRNA that is the same as 91B, then we should again call it microRNA 92B. But if it's coming from a different gene, then it's going to be microRNA 92B1 and 92B2. But because this is not the case, we have only one. Now, let's dedicate a certain amount of time to this 3P story, or 5P, as we previously see. Sometimes it's 3P, sometimes it's 5P. So let's go back to the pre-microRNA. We know that this is the structure that is produced, the transcript that is produced from a gene, and you have these marks that really shows you that there are different types of structure within the structure. And here you can see that Drosha is the one that will cleave this part in the nucleus, and then this part here is going to be what is left in the cytosol. And of course, this is called 5P prime arm because it is on the 5' and before the loop, and this is 3P arm because it is 3P prime and after the loop. But when you cut the loop by dicer, then again you will have 5' and 3' on both parts of this duplex. Now, this is the duplex that needs to be loaded on the RISC, and while it's loaded on the RISC, there is, because RISC is not one protein, it's a very powerful complex, there is an Argonaute protein that actually determines which of these two arms is going to be leading and which is going to be passenger strand, which means that one of the is going to be designated for interact sequence mRNA and the other one is going to be destroyed. What really defines this is the energy of 5' ends of both strands. The lower the energy, the less the stability, and basically if stability is lower, then that strand is going to be used by RISC to be designated as a strand that's going to target certain mRNA. Now, very interesting question was happening during the training school. One colleague from Milan, I think, asked me, so does this mean that in all the cases, in all the diseases, always the same, in all tissues, within the same scenario, the same microRNA will only give you 5P to be, for example, the targeting one, or in other scenario 3P, or this can be interchanged? And actually it can be interchanged. Sometimes it happens. We still don't know the mechanisms after that question because I really didn't know the answer why this can be different. I discussed this with Anna Yale and other people that are relatively specialized in the microRNA field, and we really discussed this in depth, and actually there is no concrete mechanisms nor information how we can understand why sometimes these things can change. There are many mechanisms that are currently being discussed that can explain why, although what I already explained to you, why sometimes RISC complex can favor the other one instead of the one that we thought might end up being the leading sequence. So to answer the question, yes, we can have different types of selection. which is not canonical, let's say, but we still don't know, or currently there is not consensus what are the different mechanisms that drives this. So the next type of RNAs are small interfering RNAs, and I put here on the slide synthetic, which is arguable because in the nature, some organisms can produce small interfering RNAs. But from my perspective, when we discuss cardiovascular disease and targeting therapies and this complete field, I would say synthetic, because this is something that we synthesize in order to target certain genes. So mostly when you discuss small interfering RNAs, and the entire mechanism behind all this is something that you want to synthesize in order to target certain genes. Everything else is more or less the same for the microRNAs. They are 20 to 25 nucleotides long, but because they are synthesized to perfectly match the targets, then there is only one outcome when you produce small interfering RNAs and transfect cells with it, or use it as a drug. What is going to happen basically, because small interfering RNA is the duplex, so you have again 5p and 3p arm. They are going to be loaded on the RISC complex, which is the same mechanisms that is used by microRNAs, and then activated RISC is going to find its target. But now, because there is perfect match between the target and scRNA, you will end up with only one scenario, and that is complete destruction. Now, the trick with siRNAs is basically how, at least in the body, when you are doing this in a dish, it's relatively easier. But if you want to use this as a drug therapeutic, then there is always a question how to deliver these siRNAs to target tissues, and several types of approaches are used. Either you load it into certain lipid nanoparticles, or you modify the sequences with different types of modifications, which will increase the bioavailability and the interaction with specific type of tissue. And one very representative example of very successful drug is inclisiran, which is a drug that is basically small interfering RNAs, and it's very important for the atherosclerosis field, because this is the siRNA that targets PCSK9, the protein that is important in the destruction of LDL receptors. So just to make long story short, we don't want to have PCSK9 if we don't want to have high cholesterol. So high PCSK9, high cholesterol, because PCSK9 destroys basically LDL receptors. I mean, it doesn't destroy it, but it doesn't allow it to recycle properly in the cell trafficking processes. So basically, more PCSK9 you have, less LDL receptor you have. So what we have here is siRNA that targets mRNA of PCSK9. So we destroy PCSK9 before it becomes protein, and then we have more LDL receptors on the membrane, less cholesterol in the circulation. Another type of small RNAs are small hairpin RNAs, which again share very similar story to the microRNAs and to siRNAs. It's just that they are being delivered as through the viral vectors or plasmid to the cells, and they are reversely transcribed to DNA, which means that they are going to be incorporated in your DNA. So your DNA of interest will acquire this specific region, and then during the transcription, polymerase will produce shRNA that will end up using the same mechanisms, the same processes as microRNAs. So you can clearly see here the difference between siRNAs and shRNAs. shRNAs have much longer effects, because you administrate sirna. Sirna has its effects, and then it's going to be destroyed. But for shRNAs, because they are incorporated in the DNA of your cell cultures or any experiments that you do, then the effect is going to be much long-lasting in comparison to sirnas. These are just some microRNAs that are important in context of atherosclerosis. I just put this representation for you here, so you can really by yourself go through this image and really see how many different microRNAs are implicated in different parts of atherosclerotic process and how they are enriched in different cell lines. And this is why when I told you that it's really hard to capitalize microRNAs on the terms of drugging them and using them as drug targets, you can really see that they co-regulate many processes here. I just emphasize microRNA 155 just to see that it's important for the migration, for the monocyte differentiation, for senescence. So many pathways are regulated. Also, when we discuss atherosclerosis, we shouldn't only look at the plaques, but also in the liver, because we know that everything, I mean, not everything, but big part of atherosclerosis can happen actually in the liver due to the disarrangements and disruptions of cholesterol metabolism and lipid metabolism in general. And you can see also that some microRNAs that are important for this are also listed here. Next part of the talk is going to be devoted to long non-coding RNAs. The field is really exploding, I think, in the past 5 to 7 years. This is just a representation of different types of long non-coding RNAs, and you can see here that they are very versatile in terms of their origin. They can be either from sense or antisense. They can be from introns, exons, from promoters, enhancers. They have many different origin in the DNA. Currently, there's an estimation that there's between 16,000 and 100,000 long non-coding RNAs. They are called long because they are more than 2,000 nucleotides long. They are not very well conserved across species, which means that if you want to analyze and functionally describe these non-coding RNAs, you can forget about animal models, because mostly in mice and men, there's a huge difference in these long non-coding RNAs, at least on the primary, in the terms of primary sequence. They have multiple targets and functions. They can exert cis and trans activity, which means that they can act within the genes where they synthesized or across different chromosomes. In trans activity, they can be active in nucleus and cytosol. You will very often read in the literature that they have very high tissue specificity, and later on I will try to argue about this. They are not very well abundant. Again, something that we can argue. We will see that. They have many isoforms. Sometimes it happened that two isoforms have completely different functions. For example, for Unreal, very famous long non-coding RNA, you will find that one isoform is pro-inflammatory and the other is anti-inflammatory. They can be druggable. This is something that is currently, I think, many teams are working on using long non-coding RNAs in drugs. They can be biomarkers again, but due to low abundancy, it's very hard to measure them with conventional methods, and at least in body fluids. So this is from Tabula Sapiens. This is single cell sequencing data. And I put here GAPDH, which is an actin beta, which are the genes that you are probably familiar with. They are housekeeping genes. And then I put some random long non-coding RNAs that are mostly associated with atherosclerosis or cardiovascular disease in the literature. And then you can really see, for example, for MALAT1, so the darker the color is, the greater the expression is. So you can really see for MALAT1, it's not very low abundant, I would say, compared to housekeeping genes, nor it is super specific, because you can see that over different tissues, there's a high level of expression. With NIAT1, you can get more or less the same, maybe a little bit more tissue specificity, because you can see that these circles are different in different Again with Unreal, this one is Unreal. You can see that it's very low abundant, but again present in many tissues. With MEG3, you can see that in some tissues is more present than less present. So when we discuss about this high tissue specificity and very low abundance, please always be careful about these characteristics of long non-coding RNAs, because this is all context dependent. Because if you go with sequencing depth of 10 million reads, you will not see many of long non-coding RNAs, and then you will see, aha, these are specific because I didn't find any others. So when you read the papers, or if you are reviewing them, please always check these things that I advise you here to do, because for sure I can tell you that for most RNAs, I mean, you will see many papers that are being published about MALAT1 measured in plasma or in serum, and I can tell you from my personal experience that this is very hard to do with traditional methods. So just be careful. When it comes to the function of long non-coding RNAs, again, they have many different functions. It's not so straightforward like with microRNAs, siRNAs, and shRNAs. They can be active, as I mentioned earlier, in the nucleus, where they can do many different things. They can really bridge the enhancers to the promoter. This is something that Dimitris emphasized previously, and this is why I changed the title from post-transcriptional to post being in the brackets, because here you can see that long non-coding RNAs are mostly active within the nucleus, really doing different things, but basically on regulating transcription of certain genes. But also some of them are enriched in cytosol, and there they can be Miron sponges, which means that they will bind microRNAs, and then microRNAs will not be able to target targeting mRNAs. So in that sense, you will change the landscape and the function or the state of the cell. They can also bind to certain parts of mRNA to increase the stability. Also, sometimes they can decrease stability. So overall, quite different mechanisms. I here emphasize just a little bit about their ability to co-regulate transcription. Dimitris already explained this story in great detail. So basically, if you want to have transcription, your chromatin needs to be relaxed, and certain type of modifications like acetylations will always bring relaxation to the chromatin. But if on the other hand, the other very important modification of histones is methylation, but with methylation it's not straightforward. Sometimes it can relax chromatin, sometimes it can block it. So with acetylation is more binary code. With methylation, it's a little bit hard to really draw any definite conclusion. Sometimes can be one or the other, depending on the gene. And this is one of the also most prominent examples of long non-coding RNAs, hot air. You can see that it is very long. It has 3 domains. One domain that binds LCD. The other one is binding the PRC2, and the middle one is interacting with the promoter of the gene. And then basically what it does, it brings 2 components together, PRC2 and LCD, and PRC2, and both of these are acting as, one is methylase and the other is demethylase. So PRC2 promotes methylation, but again on certain histones, while LCD promotes demethylation of other histones, and the combination of this methylation and the methylation basically condenses chromatin and blocks the transcription. So this is like very, very straightforward example how long non-coding RNA can go in and regulate transcription of certain genes. Again, when it comes to the activity of, or I would say role of long non-coding RNAs in atherosclerosis, there are, again, so far that has been described numerous long non-coding RNAs. Again, you will find some of them in different parts of the story. For example, on REAL, we know that it regulates smooth muscle cells, it regulates inflammation. So again, with a specificity, and we should be really, really careful with that. But basically, every day, or I don't know, monthly, we can see that another long non-coding RNAs has been described that really is important for the regulation of many steps in atherosclerosis. So the field is really growing. But again, if you read about long non-coding RNAs in animal models, be really, really careful and always check. If you, for example, want to study long non-coding RNAs in humans, people with myocardial infarction, and read cool paper about, I don't know, ApoE knockout mice that really show that this long non-coding RNAs has very meaningful function in atherosclerosis, please just first check in certain databases if you can find these long non-coding RNAs within the human genome, because for many the translation is not possible. So just be careful about it. And the third type of the non-coding RNAs that are being studied, again, relatively extensively in the past couple of years, are circular RNAs. And again, the name is pretty explanatory, so they are full circle. We know that they are abundantly expressed and highly conserved across species. Multiple targets and functions are also characteristic of this type of RNA. They say that is expressed in tissue and cell type specific manners, but again, I would be very, very careful, because if you go back to what Fabio was discussing a few months ago, or half year, or maybe 6 months ago in a webinar where he presented some results about circular RNAs, you will see that, again, you need to use very deep sequencing, very sensitive methods to discover circular RNAs. So again, with the tissue specificity, I would be really careful when it comes to stating that. They can be relatively short or long, depending on the circular RNA. And regarding what this circle is made of, there are three major types of circRNAs. It's exonic, which basically they only have exons within them, exonic intronic, which is combination, and only intronic circRNAs. When you look at the biogenesis of circular RNAs, you can really clearly see that basically they are always being produced not by specific genes, but mostly from exons and introns that are part of other genes. So it is not unusual to see a gene and a circular RNA that is also coming from the same gene. This is something that you will find in the literature, and that there are two different counterparts of the same gene. One is circular and it has its own function, and one is linear, which can be coding RNA or transcription factor or whatever. So in this case, when we focus on the process how circular RNAs are being produced, the first prerequisite, the first thing that needs to happen is basically to have this hairpin shape within the genome. Now, this hairpin shape can be formed, I would say, spontaneously, because there are palindromic sequence within the genome, and then basically when they are next to each other, they spontaneously form this type of shape of secondary structure. But in other cases, we have RNA binding proteins that are catalyzing this, forming this type of structure. And sometimes it is the lariat form that is formed during the splicing of the introns. In any case, what actually happens here is there is a promotion of connection between the beginning and the end of this structure, and that is called back splicing. And basically you have circular form, which doesn't have beginning and the end formally. So it is much more stable in terms of destruction, because we know that RNAs is always like to attack on one or the other side. And by having this completely closed circle, half-life of circRNAs is much more longer than other RNAs. So basically, yes, depending if you just have two exons that are going to be connected in circle, then it's going to be exonic. If you have combinations of exon and introns, it's going to be exonic intronic. And if you only have introns, it's going to be intronic circRNAs. These one, of course, are mostly produced during the process that is called intron splicing. You know that during the maturation of mRNA, there's spliceosome that really targets the introns, pull them off, and then seals the exons. And in this process, that there is this structure that is called lariat, and then basically out of this structure you get full intronic circRNAs. Regarding the function of circRNAs, mostly in the literature you will find that they are also, as mentioned earlier, for long non-coding RNAs, Miron sponges, which means that they can bind many microRNAs, and then basically they just diminish the effect of these microRNAs on the target RNAs, and then save certain target RNAs. But there are other functions like protein sponging, blocking translation, protein sequestering, scaffolding. They can also go back within the cytosol and regulate, again, on the transcriptional level, they can regulate the process of transcription of certain genes. So again, many different functions. Within the atherosclerosis, currently, because they are only just, the field is just being, developed or evolved in the past couple of years, there has been a limited number of circRNAs that were connected to atherosclerosis and other cardiovascular diseases. And here you can just have them listed, and you can see what are their functions. So mostly they target microRNAs, as I previously mentioned. They sponge microRNAs, and that is by far mostly studied mechanism within these type of RNAs. And for also something that really needs to be taken into account when we consider microRNAs is a new field of research. Well, we understand that microRNAs can also be endocrine regulators, because what we've seen so far, that different types of cells can release so-called extracellular vesicles, which can basically be filled with different proteins or different non-coding RNAs, mostly microRNAs, or at least they are more studied. And then these extracellular vesicles grow from one tissue to another, or from one organ to another, which is, again, something that is very similar to hormones, and then regulate, of course, when the target cell integrates this vesicle, the entire material goes within the cell, and then microRNA can come and regulate certain aspects. So this is really interesting to me, this new field, when basically you look at microRNAs as a communicators between cells. And here is just a representation for me that is very interesting to see the microRNAs that are so far being discussed as potential regulators, endocrine regulators of processes that are important for atherosclerosis. I would like to finish up with something that was to a certain extent touched by Yael in some of her webinars previously, but I just felt like it is important to also discuss the world of RNA modifications, the field of epitranscriptomics, particularly because I'm currently working in this field more extensively. So this is another layer of transcriptional or post-transcriptional regulation. So basically, if you look RNA, of course, we know the classical story about the adenine, guanine, cytosine, and uracil. But the fact is, this is just a portion of RNA world, because there are many, many different nucleotides, that more than 100 modifications of different nucleotides has been described so far. And for some type of RNAs, this is what we know. For ribosomal and transporting RNAs, we knew for ages that they are heavily modified, and we knew that these have functions within these RNAs. But lately, we have been observing the significance of this modification also on mRNA, on microRNAs, long coding RNAs, and we know that these modifications are very important, not only in the biogenesis, but also in the function of certain types of RNA. So for this one, I will just focus on the one of the most present modification, which is called N6-methyladenosine, or short M6A. And basically, if we go back here, you can see what is the modification. Here you have methyl group on this N7 position. So it's very similar to methylation of the DNA, and you are probably more aware of the process of the methylation of DNA. There you have the enzymes that can cause the methylation to appear on certain position, which are called writers because they write. You have erasers, which remove this type of modifications. And also in this scenario, you have readers. These are the proteins that can recognize these specific modifications and interact with it. In case of M6A, the writers are very complex. So here you can see the complex. It's not these, this part here is not multiple enzymes. It's just one large complex. And within this complex, methyl 3 part of the complex is the actual enzyme that that perform the methylation, but all the other parts are needed if you want to have this reaction happening. So very, very complex enzyme. Also, beside this very complex enzyme, you have methyl 4 and methyl 5 that are shown to be also employed in the modification. Methyl 4, I think Yael gave us wonderful presentation of her work, is more important for small RNAs. When it comes to erasers, those are the proteins that, or the enzymes that destroy or remove this type of modification. And in case of RNA modifications, there are only 2 so far, or these are the ones that are mostly mentioned, which is FTO and ALKBH5. Now, having this modification within the mRNA without readers doesn't mean much, because it doesn't really by itself do anything to the RNA. But when it comes to readers, these are the proteins that actually recognize the presence of this M6A on specific positions, and then it creates certain effect. So here I just list several of the readers that are currently known to be very involved with these interactions in the literature, and you can see that certain types are more, let's say, employed in the mRNA degradation. So basically, what it means, if you have M6A position on certain M6A on certain position in certain mRNA, that can cause greater degradation because of the interaction with these readers. Others are more involved in the mRNA splicing or enhanced translation. So for some proteins, if you have if the site is heavily modified, then it will increase the level of translation, and then you will get eventually more proteins. So basically, yeah, they regulate the, I would say, dynamics behind the mRNA world. So it's really, really exciting field. And we are currently working on a book of transcriptomics and atherosclerosis, and one entire chapter is dedicated to this story. And I was just reading and writing this in past few days, and I can tell you there are numerous examples of how this is related to the atherosclerosis and the development of atherosclerosis. So it is really important for you to take this into consideration when you want to analyze certain aspects of atherosclerosis genesis or progression. Again, when we discuss about the M6A, it is the most abundant modification, but according to our calculations, because we did some analysis, it's basically 0.2% of adenine is modified with this. So don't think when I say, don't let me fool you, and so you are misled that there's like all adenines in certain RNAs are methylated. No, this is not the case. Also, what I want to emphasize, previously we discussed about the microRNAs and circRNAs. You will see when you go deeper into understanding how biogenesis of microRNAs and circRNAs is happening, that methylation of certain transcripts on certain position is crucial for them to being able to form circRNAs in the case of circRNAs, and also for the transcription of or genesis of microRNAs. And with that, I would like to just remember one more time our great training school in Crete. It was really success, and thanks to Dimitris and Christos so much for this event. And really, I hope we are already preparing the training school for the next year. So at some point early next year, you should be receiving invitations, and really consider joining us and applying for this, because the atmosphere was really amazing and we all learned a whole bunch of new things. So thank you so much. That was all from my side. And of course, I will be happy to receive any questions from your side if you have any. Let me just check the chat.
There are some questions.
So much, Miron, for this wonderful overview of all the details of the multiple families of microRNAs and non-coding RNAs and so on.
I have interesting question here. Let me just read it. So Nomi, thanks us. Thank you for joining us. There is a lack of standardization tools and methodologies for studying a long non-coding RNA. Yeah, that's a great question. Such loss or gain of function techniques, which makes it difficult to study them, and also the low expression. How do you think we overcome these these pitfalls? Yeah, this is such a great question. I think we emphasize this in so many papers so far, that every paper that I read in the past 2 years, I think I always go back to comment this. To be honest, I don't really know. We need some type of consensus. First, I think the first step is that everybody is aware that long non-coding RNAs are not tissue specific. This is why I emphasize that. And also that there are many isoforms of long non-coding RNAs, so of specific long non-coding RNAs. Sometimes there's even 10 different isoforms. So when you measure them, and when people start doing this, it is really important how you design your primers. Are they, do you measure all the long non-coding RNAs? Let's start from that point, because qPCR is something that is fairly well known among all of us. So you need to design primers. If you want to target specific long non-coding, specific isoform, then you need to target this with specific primers, or you need to design primers that include all the different types. This is something that is important. Another thing is that they are really low abundant. So it really is up to researchers to have a certain research integrity when they publish results about long non-coding RNAs in body fluids, because whenever I had reviewed maybe 5 papers in the last year that were submitted, and whenever I asked for the researchers who were doing measuring of long non-coding RNAs in blood to submit raw data to see the CT values, these papers were not, they were never resubmitted for the review, I can tell you that. So this is another thing. From my experience, when we were measuring MALAT in plasma or even in PBMCs, I mean, not in PBMCs, but in plasma from patients, even if it's very highly concentrated RNA, I mean, we used large amounts of plasma to isolate. It was like 36 CT. That was the CT value for MALAT1, which I showed you, that is one of the very, very highly expressed long non-coding RNAs. When it comes to discovering new long non-coding RNAs and more about their functional analysis, this is something that I'm not really expert on. I can only say that I see that people, when they want to discover new long non-coding RNAs in specific organs, they go very, very deep with the sequencing, like 200 million reads, 400 million reads. Again, on the that really, for me, from the biological perspective, if you have long non-coding RNAs that does something but is really that low abundant, does it really make a difference biologically, and does it make sense to biologically analyze it? I don't really know. Another curious fact is that we, as I mentioned, we cannot really use animal models that extensively to validate functionally analyze these long non-coding RNAs. But this is something that really struck me. I read this in one paper or two papers, that although primary structure of these long non-coding RNAs, it's not the same, their tertiary structure is relatively similar, and when it comes to long non-coding RNAs, and at least when we discussed about their roles in the cytosol and how they regulate transcription, it's much more important. 3D shape is much more important than what is their primary sequence. So this is another, I would say, niche where the field needs to get more insights and to really try to get new tools for 3D programming or getting this information, because it would be really nice to see comparisons between, for example, mouse 3D shapes of long non-coding RNAs and humans, and then we can maybe find more connections than just going into primary sequence. So I know that I didn't give you much of information. Maybe I just kind of open new doors, but basically this is what I envision as a problem. Also, why is long non-coding RNA expression so different between human and mice, for example? Well, as I showed you in the beginning, what we know is that the complexity of the organisms is really hidden in the dark genome and within these long non-coding RNAs. They are quite different, and why there is no sequence conservation, I cannot really understand this. I don't know, but as I mentioned, even if the primary sequence is not the same, the 3D structure are relatively similar, which means that this should be really emphasized and analyzed from that perspective, not just analyzing the primary sequence. Okay, do we have other questions? Yeah, I understand your doubts because you are more microRNA focused. No, I'm just kidding. But yeah, also when long non-coding RNA field exploded, I was so into it. I really wanted to go that way, but as you also, I hit so many walls in from the methodological part that I'm also doubting that these first publications, that showed us the potential. I can for sure see limits in the field currently, but with new technology, we might unlock this potential and really move the field forward. Okay.
Miron, can I ask?
Yeah.
So I have a question about the circular RNAs. First of all, how abundant are these? Are they abundant in the cell? Abundant.
Yeah. Again, this is the same as with long non-coding RNAs. Some are more abundant, some are less abundant. I cannot say that there is a universal answer for that. But some of them are really, really highly present. And if you go back to Fabio's lecture, remember that when we had discussion, I asked him, okay, if they are, if we really need to go this deep for sequencing, you know, what is the biological relevance of such low abundance? But then he explained it. The reason why we need to go really deep is not related to the concentration, but basically to the pipeline, how we do sequencing for circular RNA, which is a little bit more complicated than for regular sequencing.
That was my next question about about the identification and the sequencing. So I think it's not so straightforward to sequence this circular RNAs.
No.
How can we discriminate between the regular? Because these RNAs are non-coding, right? Or not?
Yes,
So they're non-coding strands. So if you sequence it, yes, from that gene, because they come from exons.
Sometimes from introns, sometimes from exons. Again, there are
from
different.
Exons, yes. If they are non-coding strand, okay. But if they were a coding strand, it would be. But if they are non-coding strand, the non-coding strand, then in the mechanism you did not, let's say, include the auto-regulation of the gene, because if this is non-coding strand, then this non-coding strand can go to the coding strand and act like an antisense, right? So, this is one mechanism, another mechanism that these circular RNAs could work.
For now, I really didn't found any information about them really acting on the gene they are coming from. Mostly what is described is in literature, and you can see this on slides, is that they act as microRNA sponges. Now, are those the microRNA that are coming from same introns or from different introns? This I don't know. I'm not really expert in circRNAs, but this is the mostly described mechanism that I see in literature. It makes sense what you said. The interaction between mRNA and its counterpart circRNA. I didn't find this in any papers. Again, not expert in the field. Maybe Fabio will enlighten us at some point with that one. But I really encourage you to go back to his webinar, at least to go and see how he described how you create primers, because even if you want to do regular qPCR, the primers should be designed differently than for regular RNAs. There is a certain philosophy behind how it should be done. So
I'm asking this because for me it's difficult to understand how an RNA is made with the specific role to be a sponge. I mean, I don't know. It doesn't make sense to me to make an RNA to act as a sponge. I mean, a sponge, this is a very general mechanism. It's not specific at all. It's not specific. I mean, sponge what?
I agree. I agree completely.
So there must be some more specific mechanism about these circular RNAs, which are, and I think it's very interesting, and it's a very new field, right? It's completely new.
I think in the past 3 or 4 years, there is really like a progressive number of papers that are being dealt with this subject. But again,
there is another question in the chat from Nicholas.
That is is it correctly understood that it is the less stable part of miRNA that will be guided strand in the RISC complex? The one that has that 5' of the so we have 5p arm and 3p arm, and both have 5' end. And which microRNAs has less energy or less stability in this 5' end, that one should be chosen to be within the RISC complex. I don't know if I was clear when I was previously discussed, but this is the how it's explained. Again, emphasizing the fact that sometimes we have exemptions to the rule, so it's not always straightforward. But generally speaking, it should be one that is less stable. That one is going to be chosen to be within the RISC complex. Okay, no more questions so far.
Here, I have a comment and a question. You mentioned the issue of the tertiary structure for clearly interaction. So this may be also important for the sponge effect.
I think not so much for the sponge effect as much as for the interaction with other proteins like scaffold function. So because 3D structures to me are mostly important when you see what is happening in the nucleus between the histones, you know, the interactions between the proteins, everything what Dimitris explained with the zinc fingers domains and DNA, RNA interacting proteins. For me, that is more, I see there greater influence of these 3D structures than in sponging, because in sponging, if you have revealed the sequence that is going to bind certain RNA, then you know it doesn't matter if it's circular, if it's like that or that. You just need the interaction.
Yeah,
it will match. It will match. Yeah.
Okay. Clear.
Okay. If there are no more questions, I would thank everybody for sticking for us too long. I hope it was interesting for you. Also, I just want to announce that the next webinar will be sometime in the end of the November. We still didn't specify it. Probably focusing more on RNA sequencing methodology in general. And also on the 18th of November, we prepared another journal club chats. This time it's going to be focused mostly on how to build your career in academia. So myself and Brooke, which are Maria Curie Postdoctoral Fellows, are going to, in 5 minutes, present what this fellowship is all about and what would be the best approach to get it. And then we will discuss more about potential jobs in academia that are not only to build yourself to be PI, but also to do other things. So for younger people, it should be younger colleagues that are YRIs, it should be interesting. So please disseminate this information. And if they can join, it would be really nice to have like a collaborative environment so we can discuss and answer questions. And then in December, we will skip the journal club because of the holidays. And then from January, we will continue with, again, sessions on specific papers. But this is like every 6 months, we organize GC chats just to relax a little bit with these more socially important subjects. So from that perspective, just join us. We will disseminate the information about the journal club. And if you have any interest, please join us.